Now, we live and breathe every day in a cloud of viruses, but most viral infections go completely unnoticed. In some cases, viruses can cause disease, and in more rare cases, they can cause an extreme amount of damage and devastation around the globe. Now, viruses are extremely tiny and deceptively simple, but looks can be deceiving. Even though viruses are so simple, we don't even consider them alive. They can evolve genetically. And so this is why in the Sagan lab, we want to understand how something so seemingly simple can do so much. Well, you've probably heard of DNA before. You can think of DNA as the library where we store all of our genetic information. And when we want to take a gene off the shelf and make something from it, what we do is we copy that DNA into RNA. Now, RNA is therefore thought of as the messenger between DNA and proteins. But RNA is so much more than just the messenger. RNA, like DNA, can store genetic information. But like proteins, RNA can fold into complex three-dimensional shapes and structures, and actually even perform chemical reactions like proteins can. And so in the Sagan lab, we study RNA viruses like Hepatitis C virus, Dengue, and Zika virus. And these viruses, their genomes are made of RNA. And so they use that RNA in order to take over our cells and produce more viral genomes. What we really want to understand is how viruses use their RNA to do this. How do they use their RNA to make the viral proteins? What signals on the RNA allow them to copy their genomes? And how do they turn our cells into factories to make more infectious viral particles that can go on to infect new cells and new hosts? Ultimately, what we really want to do is take the knowledge we learn about how viruses work and use it against them so that we can stop the cycles of damage and devastation.